Welcome to episode 3 of the podcast for the Journal of Neuromuscular Disease. I'm Grace McMacken. I'm a trainee neurologist, and today I am absolutely delighted to have 3 experts joining me to discuss the topic of induced pluripotent stem cells, and more specifically, how we're using these to study neuromuscular disease. So I have with me Professor Frederique Magdinier, who is leader of the Marseille Medical Genetics Research Unit, which is jointly affiliated with INSERM and Aix-Marseille University. I'm also joined by her esteemed colleagues, Dr. Leslie Caron and Dr. Stefano Testa. We really wanted to bring you on the podcast following on from your really interesting and very comprehensive review article, which was published in JND in September 2023, so volume 10, issue 5. And it's titled Induced Pluripotent Stem Cells for Modeling Physiological and Pathological Striated Muscle Complexity. Listeners can find the link to that in the episode description if you're listening on Spotify. And like all of our articles in JND, it is, of course, open access. So thank you so much for joining us today, guys.
Thank you for the invitation.
Thank you.
I want to maybe direct my first question to Stefano. In vitro modeling of cells can be a really tempting prospect for researchers, but muscle can be a bit tricky. Can you tell us a bit more about what makes muscle difficult to model in vitro?
Okay, yes. We have to take in account 2 different levels of complexity. The first is the biological complexity, so the cells that create the muscle, and the second is the organization of the muscle, so the organization of the element. Starting from the cells, in muscle, we found a lot of cell population that lives together in the same organ, in the same tissue, maintaining the homeostasis and also allowing the functionality of this tissue. For example, we have the muscle cells, of course, the myoblasts, that are very particular cells because are single cells that, when start to differentiate, fuse together to form a sort of society of cells, that is the myotube, that is a structure with different cells share the cytoplasm and the membrane. Then, for example, we have not muscular cells that take part in the developing of the connective sheets, that are these tissue that surround the muscle fibers at a different level. Then we have, for example, the blood vessels, so cells, angiogenic cells in the tissue to create these blood vessels that are essential for the nutrient supply. Very important in the muscle is the innervation, so there are nerves inside the tissue. And of course, an important role have also the immune cells and, for example, the macrophages, that plays a very important role during the regeneration of the tissue. And so this is a little the niche that it's present in the muscle tissue and that it's quite difficult to reproduce in vitro. The other aspect is the organization of the muscle, because this tissue is organized in a hierarchical way. So starting from the lower level, we have the muscle fibers, that is surrounded by a very thin connective sheet called the endomysium. Then more fibers together forms a bundle surrounded by the perimysium. And then more bundles together forms the real muscle surrounded by the epimysium or fascia. And this sheet is connected with the tendon. So it's important to make the link between the muscle and the bones. And finally, all these muscle elements have also to be aligned on the same direction, because when we have the contraction, the muscle have to be shorter to become shorter. And so all these elements have to be very well aligned to have an effective contraction. So for me, these 2 main aspects are the bigger difficulties in modeling the skeletal muscle tissue.
Thank you for reiterating some basic muscle physiology for us as well. That's always really important to to have fresh in our minds. Stefano, can you tell us at the minute, what are some of the ways that researchers are modeling muscle in vitro?
For sure, to model the muscle, we need a good cell source. So a good cell type that have a myogenic potential. The myogenic potential is the ability of the cells to differentiate in muscle. We can use, for example, primary cells that are just isolated from biopsies, so from a tissue, and are cultured like this, so without genetic modification. So these kind of cells, so an application in human, are the best cells because are not genetically modified. And for example, we can isolate stem cells, so primary stem cells, in different tissue of the adult body. So we don't need the embryonic stem cells, but we have adult stem cells in our body. For example, in the muscle, we have the satellite cells that are the real stem cells of the muscle. But we have also myogenic cell population in the perivascular compartment that are like the pericytes and the mesangioblasts. Then we can isolate stem cells with myogenic capabilities also from the adipose tissue and also from the bone marrow, for example. So the good things of these cells is that are untreated cells, so are naive cells, but the limitation of these cells is that it's very difficult to keep them in culture for a long time, because they, in a few days, they start to go in senescence. So we have a very limited number of duplication that these cells can do before stop growing. So this proliferation, this limited proliferation, is a problem because if we need a lot of cells to create a tissue, we cannot use this cell source. So one strategy to overcome this limit is to immortalize these cells. Immortalize the cells means that we modify the genes involved in the aging, so in the senescence of the cells, in order to extend the lifespan of the cells. So, for example, usually use the immortalized myoblasts. So we have a cell source that can proliferate in theory as much as we want, and then when we change the culture conditions, so when we push these cells to differentiate, we obtain a muscle. And then, of course, there are the cells that are the topic of this podcast, that are the induced pluripotent stem cells. But my two colleagues are more expert than me on this topic and will talk better about the nature of these cells during the podcast. And the other things that we have to keep in mind if we want to model the muscle is how to organize these cells. So also in this case there are different level of culture system with increasing complexity. So we can start from the classical 2D culture, so we can just culture these cells and make them differentiate. That is good if we want to study, for example, the gene expression. But if we want to study the morphology of the tissue, we need something a little more complex. So we can pass to the 3D cultures, and there are different 3D culture systems. There's the organoids that are the little one. Then we can increase a little the size with the 3D culture system that involve the use of biomaterials, so of a scaffold to keep these cells, to organize these cells in space. And for me, the best condition is the technique that I use is the bioprinting, because with the bioprinting we can not only have a three-dimensional tissue, but we can also organize the element inside this tissue.
And Leslie, can you tell us a bit more then about induced pluripotent stem cells? What are they and how are they derived?
So, human-induced pluripotent stem cells, or iPS cells, are reprogrammed cells that have hallmarks similar to embryonic stem cells, which means that they can proliferate indefinitely, and they are pluripotent, which means that they can basically give rise to any cell types of the body. So unlike embryonic stem cells that are naturally found in the early embryo before its implantation, iPS cells are reprogrammed in a laboratory from adult somatic cells that were already specialized. So the reprogramming is a process that transforms a cell into an unrelated cell type. And in the case of iPS cells, specialized somatic cells reacquire prepotency through a global resetting of their biological clock. So this process of reprogramming allows them to go back to an original undifferentiated or unspecialized state. So they reacquire their full potency of becoming any cell of the body. So once they are reprogrammed, iPS cells share several characteristics common with embryonic stem cells, including their morphology, the expression of prepotency markers, and the ability to differentiate into virtually all cell types.
Okay, thank you. And why would we use these over the other cell models that Stefano has discussed?
Of their very important characteristic is that they can proliferate indefinitely in vitro. And Stefano mentioned earlier that usually cells, when they are cultured in vitro, they enter senescence, and we are very limited in what we can do with them in the laboratory because they start dying, basically. So these iPS, or that's the same for embryonic stem cells, in vitro, we can keep them forever. They will not enter senescence, so we can keep them proliferating. So basically, we can generate a huge amount of iPS cells. So they have become a very valuable tool in research because this characteristic, in combination with their ability to differentiate into any cell type of the body, allow us to generate a huge amount of specialized cells. So we can study specific tissues like muscle cells, for example. And no other cell type allows us to have such quantity of cells to work with in a laboratory. So that's one of their specific interests. And another one is because they are pluripotent and they can differentiate from basically a very naive and differentiated state to specialized cells, we can study all steps of the differentiation. So to basically recapitulate what happened during embryonic development. So that's also culture systems that allow us to study normal or abnormal human development. And most of the knowledge we have on human development was extrapolated from study made on other models organisms. Most commonly used are rodents or flies. But as you can imagine, those animals don't always reflect human biology. So it's important to develop tools that allow us to study human development. And iPS, or embryonic stem cells to some extent as well, allow us to study this different state of embryonic development. Another benefit of these iPS cells is because it can be derived from individuals, as they are derived from individuals. They can be derived from patients that carry mutations or have some disease, genetic disease. So we basically end up, like after reprogramming, we end up having cells that carry the same genetic disease. So we can study these cells and we can model this particular genetic disease in vitro.
Wonderful, thank you. And so specifically, can you tell us a bit about what methods are currently being used to differentiate iPSCs into skeletal muscle?
So originally, it was very difficult to make skeletal muscle cells out of iPS cells for a known reason, I would say. This cell type was not easily achievable. And like other cell types, like neurons or cardiac myocytes, it was very challenging. And it took decade for the first laboratory to be able to make skeletal muscle cells out of iPS cells. And initially, the first protocols use a forced expression of skeletal muscle markers, specifically MyoD or PAX3, PAX7, markers of skeletal muscle progenitors or myoblasts, to push the cells toward the muscle path. But for some, it's not really practical for some experiment. It's because it can lead to modification of cells. So several laboratories have worked on developing other protocols and that don't require the use of this forced expression. And now there is about 10 protocols that have been developed and that don't use transgene. And this protocol basically involved the use of combination of small molecules and growth factors on different steps. And these steps will recapitulate, mimic the development of muscles in vivo. So we try to mimic as much as we can the development of the muscle to recapitulate that in vitro. And I would say it works quite well. Now there are a few protocols that are pretty efficient and easy to use. We have two in the laboratory. Some protocol will lead to the generation of a pure population of skeletal muscle cells. Others are based on the co-differentiation of iPS into muscles and motor neurons. These motor neurons are important to activate the muscle because they innervate the muscles. In those protocols, usually muscle cells are a little more mature, and there are also protocols that lead to a 3D structure that we call muscle organoid. So this structure recapitulates muscle tissue. It's not a single population of a particular type of cells, but it's a mix of several populations that are well organized, and those populations are the ones that we find in the human tissue, in the body.
Frederique, what models of neuromuscular disease exist using these human-induced pluripotent stem cells?
So as Leslie mentioned, the field is a bit lagging behind compared to other diseases because it took a long time for researchers to set up good protocols to make muscle out of iPS, human iPS. It was easier in the mouse, but human iPS, for some reason that we don't really understand, were kind of resistant. So of course, in our last survey that we did during the writing of the publication and the review that we published so in JND in September, the most common type of disease that was modeled is Duchenne muscular dystrophy. So it's been modeled by different means, by overexpression of myogenic markers, but also by using different cocktails of small molecules. And there are many publications that report the modeling of Duchenne muscular dystrophy for several reasons. First, of course, it's the most, let's say, common type of muscular dystrophy. Second is because a lot of teams are working on this disease and try to find a cure for the patient by doing different type of testing, like pharmacological treatment or gene delivery or even cell therapy for muscle regeneration. But then a few other diseases have been modeled using iPS, like I can mention limb-girdle muscular dystrophy, we can mention myotonic dystrophy, we can mention facioscapulohumeral dystrophy, myocardial disease more recently, and also some disease, neuromuscular disease have been modeled, but more on the neuromuscular side, on the motor neuron side, like SMA, for instance. But this was mostly based on the differentiation of iPS into motor neurons. And then sometimes some co-cultures between motor neurons derived from iPS and then muscle cells, like primary muscle cells or immortalized myoblasts, as Stefano mentioned. So the number of diseases remains limited, but I think this will expand in the near future because now protocols are more efficient and viable more largely to the community.
Oh, great, thank you. And I suppose there's no perfect model, but do you think that these models are as relevant to the human disease as, say, animal models?
So the good thing with the iPS cells is that you can get the cells from the patient, and for a single disease you can test a whole bunch of different mutations, which is not really feasible in the mouse because it's more complicated to make mouse models, and you cannot really model all the different mutations for a single gene. So this is a big advantage because it's cost effective. It's not cheap, but it's cost effective relative to the production of mouse models. It's also good because these cells can be used for different type of things. So Leslie mentioned the characterization, but we can also use these cells. You can also use like cells. We can also set up a number of readouts. We can use different techniques to stimulate the cells and then identify a readout that can be used for drug discovery, for instance, or for therapeutic testing before going to animal models. So for screening issues and for screening purposes, I think they are very, very useful. And also, it's going probably to be a very good mean to identify new genes and new mutation because a number of patients still remain without a diagnosis. And of course, if we don't know the genetic mutation in this patient, or if we have different mutation of unknown significance, then it can be modeling this or testing the mutation in this patient using iPS directly derived from the given patient. Is very useful to do pathophysiological analysis before eventually going further and making mouse model. So I think there is a great potential of using iPS at the moment, especially for neuromuscular disease, for sure. Yes.
Excellent, thank you. And Leslie, as Frederique has alluded to, there are some ways that these can be used in throughput screening for therapies. Can you tell us a bit more about that?
First, iPS are very good tool for regenerative medicine and cell therapy in particular because again they can be differentiated in any cell type of the body and they can proliferate indefinitely. We can make huge amount of these specialized cells, and this is what is required for therapy like cell therapy. We normally inject big amount. This is what's necessary to have an effect in the human body. And that's not possible with most of the cells that don't easily proliferate. So that's one of the aspects, and because they can be derived from the patient, they are genetically identical to the patient they come from, and then they will not trigger an immune response when they are transplanted. They have kind of revolutionized cell therapy. And this type of cell therapy approach involving iPS cells are currently being tested for Parkinson's disease, for example. And Parkinson's disease is a neurodegenerative disease where dopaminergic neurons deteriorate. And there are currently several clinical trials involving human embryonic stem cells or iPS cells to differentiate into dopaminergic neurons that are transplanted in the patient and to test their safety and efficiency. So I think the results are not published yet, but they are very promising, and we can be confident that such strategy will be developed for other disease and potentially muscular disease. But that is not the only way human induced pluripotent stem cells can be used for therapy development. We can use them in vitro as a human preclinical model to test innovative therapies. So as of now, about 85% of preclinical tested drugs will fail in clinical trial. And the reason is because most of these drugs have initially been tested in animal cells or animals. And because animals don't always reflect human biology, that mislead the development of this drug. And this is where iPS have also revolutionized medicine, it's because there are now iPS that carrying, for example, genetic mutation and that are associated with disease. So they are used as a platform to test molecules that could have an effect and revert the deleterious effect of the disease on a particular cell type, shorten the time of drug development because that leads to molecules that we already know will work on human cells. And they are better at predict their effect on patients in clinical trials.
Okay, so it sounds like a very promising model, hopefully for lots of exciting therapies. Frederique and Stefano, can you tell us about some new approaches on the horizon in this field?
Yeah, I think that if we combine now all the technologies that have been set up for muscle that Stefano mentioned at the beginning, and then we apply all this technology to iPS, then we will have a very large, let's say, toolbox to really model muscle in the field. There is a great it's greatly the field of bioengineering is greatly improving at the moment, and also genomics is also providing very interesting approaches and tools, and 3D modeling and organoids is also a clear revolution in the field of cell biology at the moment. So I think that in the near future we'll be really able to have like mini muscle in vitro that will combine all the features and characteristics that Stefano mentioned at the beginning of the recording of the podcast, with all the different type of tissue and somatic cells, together with motor neurons, together with muscle cells. Hopefully, at some point, we'll be able to have blood vessels and have some immune cells in our construct. And then we will be able also by using very new techniques in microscopy, where we will be able really to look at the tissue in 3D, to have to see all the cells interaction, follow the development and the differentiation of the cells, the differentiation of all the different type of cells all together, and this will be really a very powerful tool to understand muscle function, and of course also muscle dysfunction, and test some potential therapeutics for a number of diseases. So I think we are really at the bottom, and we have a huge way to go, but very, very promising approaches and development in the years to come. And it's going very fast at the moment, so I'm sure that we'll have plenty of very good tools to model this tissue. But maybe Stefano, if you want to.
Yes, I can. My expertise is in bioprinting. So currently I work with a 3D bioprinting technique based on an extrusion system. So briefly, the things that I do is I mix the cells, the biogenic cells, with one biomaterial, obtaining the so-called bioink. This solution enters liquid in the machine, in the bioprinter, and when it goes out, is a gel, is a thin filament, printing filament, in which the cells are already inside this filament with the biomaterial outside. And the good thing is that we can collect this filament to reproduce the shape and the dimension that we want. So in my case, I want to model muscle. So I collect these fibers in parallel lines, layer by layer, to reproduce the three-dimensionality of the muscle tissue. And after 1 month of differentiation, we are able to obtain little human muscle tissue in laboratory, well differentiated and very well organized. And another field where I worked was the field of the microfluidic. So basically, the things that for skeletal muscle can be important is to model neuromuscular junction. So the point in which the motor neuron take contact with the muscle and form this junction. That is very important because there are pathologies that are correlated to this structure, but it's difficult to study in the patients or in the animal because it's a very peculiar structure. It's very difficult to reproduce in laboratory, and the potential of the microfluidic combined with the IPS can have a microfluidic device where we have different chambers, so 2 separate chambers connected with a very little channels of the order of micron. So channels that are too little for the cells to be passed. So we can keep these 2 cell population separated. And for example, in these 2 chambers, we can culture from one side the muscle cells, and from on the other side the motor neuron progenitors derived from IPS, because it's currently is the only way to produce motor neuron in a laboratory. Almost impossible co-cultivate these cells together, but keep them separated. We can differentiate them in muscle and in motor neuron. And the good thing is that the motor neuron can produce the axon, so can elongate its body inside this channel and go to take contact with the muscle. So we can keep these 2 cells population separated, but also in contact. So we can reproduce the neuromuscular junction.
Fascinating. You might just need another chamber for some Schwann cells, maybe.
Yes.
So, well, I think you 3 have successfully convinced me that this is an area on the rise. And if listeners want to find out more, I think your review is an excellent starting point and a really amazing overview of what is a massive field and is clearly expanding very rapidly. Thank you so much for your time. It's been really delightful to talk to you all.
Thank you.
Thank you.
If you enjoyed the podcast, you can read the full paper and lots more in the Journal of Neuromuscular Diseases. And to keep up to date, you can follow us on social media and subscribe to our newsletter.